this special talk in Epilepsy Grand Rounds, GW Epilepsy Grand Rounds, partnership with epilepsy.com. Our speaker today is Dr. Adam Hartman from NIH. Adam is an old friend. We happened to do our fellowship together at Hopkins. He grew up in Chicago, and he went to undergrad and medical school at Northwestern. And then he did his general pediatric training at the National Capital Uniform Services near Walter Reed before he joined the Navy to be a general pediatrician for 5 years, eventually becoming the chief of general pediatrics at the Naval Hospital in San Diego. He then joined Hopkins to do his pediatric neurology and clinical neurophysiology pediatric epilepsy fellowship. And during his training, he did exquisite research at the epilepsy section of NIH, of NINDS, with Dr. Mike Rogowski, a previous speaker in this same series. And he published great papers, and eventually he stayed at Hopkins as faculty, and he earned a career development award from NIH to further his research on the ketogenic diet. And he eventually rose to the rank of associate professor at Hopkins before he joined NIH as program director in the Division of Clinical Research at NINDS. Adam has talked about the ketogenic diet in all kinds of national, international occasions you can think of. He's published a lot on the subject, and he is an expert in intractable epilepsies and their surgical treatment, including ones with inborn errors of metabolism, Rasmussen's encephalitis, other bad epilepsies that require hemispherectomy or ketogenic diet to get better. Without further ado, my friend Adam Hartman.
Well, thanks a million, Mohammed. Can everybody hear me okay? Okay, great. Well, it's a real pleasure to be here. It's always fun to see Mohammed. You know, he's a scholar, he's a clinician, he's a poet, as you all know. And this morning, you know, I always learn something new about Mohammed. I found out he's also a fisherman, so you learn something new every day. And so we're going to talk about some work that I started back when I was a resident, as Mohammed said. And so I hope to get the residents and medical students thinking a little bit about how you would sort of outline and map a bit of a career. So the title of the talk is Metabolism-based Treatments for Epilepsy, and the subtitle is How a Search for Mechanism Turned into a Path to Discovery. Just to remind you, there's 2 events coming up. One is the 2017 Epilepsy Board Review course, and if you didn't get enough today, you'll see me on Thursday and Saturday, where I'll be talking about the diet. The course is, as you all know, a phenomenal course. It certainly got me ready for my boards. And it's starting Thursday, Friday, and Saturday. And then there's also the Epilepsy Grand Rounds archive lectures on epilepsygrandrounds.com. Disclosures are listed here. I don't have any relevant commercial relationships. All right. At the conclusion, hopefully you'll be able to identify metabolism-based options for treating medically intractable epilepsy. You'll be able to quote the percentage of patients whose seizures decrease during treatment with the ketogenic diet, and you'll be able to list 3 proposed mechanisms for how the ketogenic diet exerts its anti-seizure effects. I'm not going to make you spit back and memorize the work that I've done in the lab. And here's the accreditation and CME credit slide. You get 1 hour for this, if you can believe it. Live participants, you get it on the sign-in sheet, and then if you're on the web, you'll receive a link and session code for the evaluation. So we're basically going to move through some basics about seizures. I know everybody here knows it. I want to make sure that everybody online is also starting off from the same, we're all reading off the same sheet of music. The problem is the inadequate response to medicine for many patients, as you know. And one solution is nutrient-based therapy, and that's what we're going to spend the bulk of our time talking about. So just so we're all on the same page, about 3 million people in the US have epilepsy, which we'll define as recurrent unprovoked seizures, both in humans and in rodents. It affects approximately 1% of the US population at any given time. And if you put this into perspective, so at NIH we think a lot about the scope of a problem, and when you think about the scope of epilepsy, the 1%, it's a little bit shy of 1%, is more than cerebral palsy, multiple sclerosis, ALS, and Parkinson's disease combined. The prevalence is increasing for all ages, including You've probably seen the slide a million times or variants of it. This is the Quantum Brody slide published a number of years ago, where you have, after the first 2 drugs, you have this medically intractable population where there's a very low but definable response rate. Looked at another way, and I actually almost like this slide better, was some additional work that was published in 2012, is that you have this, you sort of think of it, I hate using pie graphs, but in this particular case I think it's useful. You have patients who easily respond, and no matter what you give them, they're going to do well. You have another group in B where they're seizure-free after 6 months, so it takes a little bit of work, but they get there. And then we have the group that I'm most interested in and spend most of my time on, and that's the groups where there's either limited efficacy or there's a complete lack of seizure freedom. Ann Berg has done some work looking at what happens in pediatric populations, where you see this, some patients again go into pretty easy remission, and other patients have these remissions that are somewhat prolonged, but they keep relapsing along the road. So what do we do for those patients? Surgery only accounts for a small percentage of that piece of the pie. I'd estimate it's about 10% roughly. And surgery is an important option, and as we think about how, this is the way I sort of frame this, if someone's seizures are well controlled, then that's great. We sort of just continue medication and surveillance. If their seizures are not well controlled, then the next question in my mind is this person a surgery candidate? And my definition of surgery candidacy is actually pretty simple. If I can look at an MRI with a surgeon, trace my finger around something and say, I need you to take that out, that person's at least a candidate for surgery. And obviously, as everyone knows, there's a whole extensive evaluation to do after that. But if I'm sitting there pointing to 5 spots, then that's probably not someone who's going to do that well. So if they are a candidate, surgery evaluation. If not, then we have 3 major categories of options. We can always add more medications and in different combinations. There's neurostimulation, which has really seen an explosion in recent years. And then there's metabolism or nutrient-based therapy, which is what we're really going to discuss today. So the idea that metabolism can be used to treat epilepsy dates back to the time of Hippocrates. Some of the more senior faculty members may appreciate the name of the senior author is always the one that gets the press attention. And so it's not quite clear that Hippocrates himself really came up with this. It may have been one of his bright young students or residents. And so for the students and residents, hang in there, because it actually has gotten a lot better since the time of Hippocrates. The observation was that fasting improved seizure control. The ketogenic diet, which we're going to spend a lot of time discussing, was designed to mimic fasting, and this is actually a really important point. So just to underscore it again, the ketogenic diet was designed to mimic fasting. Okay? We're going to come back to that point. There are other diets that are used. One of my partners, Eric Kossoff, has spent a lot of time talking about the modified Atkins diet. Our colleagues up in Boston, Elizabeth Thiel, has worked on the low glycemic index treatment, and Heidi Pfeiffer up in Boston. And so there are a lot of different ways you're starting to see that altering dietary intake may influence seizure control. And so the basic biochemistry of the diet, this is one of the few biochemistry slides. When I was a medical student, I swore that I would never do anything having to do with biochemistry. I didn't understand where all the arrows went. ATP just sort of, boom, just appeared. It's like, how did that happen? And now here I am doing this. So just to sort of walk you through this very briefly, and this is again meant to be sort of a 35,000-foot view. People who are on the diet ingest fats. And those long chains, the classic diet is a long chain fatty acid diet. There's also a medium chain triglyceride diet, which we'll talk about in a minute. So they ingest fats, which then get transported into the cell. You have the CAT transporter, then the fats undergo fatty acid oxidation. You start knocking off 2 carbons at a time. Those get converted eventually into acetyl-CoA, which then gets converted into acetoacetate, which then either spontaneously decarboxylates into acetone or stays as acetoacetate, or is in equilibrium in a ratio of about 1 to 3 with beta-hydroxybutyrate. These are the so-called ketone bodies, which your body produces when you fast. Again, coming back to that fasting idea. And then something happens. And so back in the good old days, which weren't all that good, people named things and studied things based on the technology they had available at the time. And so when the diet was named, people were measuring the amount of ketone bodies that were in people's urine when they were doing fasting. And so that's how the diet got its name. It is not meant to imply that the diet exerts its effects through actions of the ketone bodies, although we're going to talk about how that area of research has really evolved in the last 10 years. So this all occurs in the mitochondrial matrix, then these ketone bodies are transported out of the cell into the bloodstream, filtered through the kidneys, and so they're in the blood, they're in the urine, and that's how the diet got its name. So how do we know that the diet works? Well, it's not just because John Freeman said it worked, it's because there have been some trials. There have been 2 randomized trials. One of them was blinded. I'm going to talk about the one that was not blinded. This is done by Elizabeth Neal and Helen Cross and their colleagues at the Great Ormond Street Hospital, where they had a cohort of patients who they identified and randomized into getting either the ketogenic diet immediately or waiting the typical 3-month waiting period. So a lot of centers, most centers, have a waiting period of 2 to 3 months before, so your physician says you're a candidate for the diet, and now you can join our waiting list for a couple of months. And so what they did in this trial was shunted a cohort of patients to go straight on the diet. You're coming in next week, we're starting the diet. So you have a natural experiment, if you will. And what they showed that there was a greater than 50% reduction in seizures, as outlined in the red box, after that 3 months. And then everybody was put on the ketogenic diet, and then they followed those patients along. The interesting thing about the control group is that it wasn't a group where things were static. They actually were changing anti-seizure medicines during that time to try to optimize their control. So in a sense, the deck was sort of stacked against the ketogenic diet, and you still see the outcomes that they had. So it's about 70, a little over 70 patients in each arm. So it's not an inconsequential number. They had the power to show the difference that they showed. So why should we explore the ketogenic diet's mechanism? So it's for a few reasons. One is to optimize the efficacy of the diet. If there's something that we're doing well, we should try to do it better. We also, the ketogenic diet, some families say, well, you know, we're interested in doing this because it's natural, there are no side effects. It turns out there are some adverse consequences to the diet. It's not that much different than medicines, although the side effects are a little bit different. But we'd like to minimize those adverse consequences if we can. We'd also like to increase the convenience of the diet. If we can make something easier, we'd like to do that. That may both overcome challenges with implementation and possibly improve accessibility to people to be able to get the diet. When you do the diet in its full form, it requires actually, you know, probably the most important person is the dietitian, and then you have to have a physician who actually knows what they're doing as well. And so it's a pretty intensive diet, but there are other diets we've discussed that are a little bit less so. Nonetheless, we want to make it a more convenient diet. We also, from a scientific side, would like to improve our understanding of epilepsy. What is it? Why does metabolism change seizure susceptibility? And if possible, identify new anti-seizure drug targets. So here's an overview of some proposed mechanisms taken from a chapter by Susan Messino and Zhong Ro. And I guess I want to point out just a few things. We're going to start off by just explaining a little bit about sort of a reductionist approach. So you have high fat, low carbohydrate. And so we'll start with those 2 factors and then sort of navigate around this map just a little bit. You can see there are a lot of arrows going in different directions. There are a couple of bidirectional arrows there. So one school of thought is that reduced glucose impacts norepinephrine and also may impact adenosine, which can activate adenosine A1 receptors, which can have an impact on KATP channels. Turns out those experiments were done in animals that were either lacking receptors or lacking the ability to produce norepinephrine, and both appear to be at least necessary for the diet to work. You also have the impact of the ketone bodies on vesicular glutamate transporters. They seem to displace some of the glutamate. There are a lot of, so since that slide was produced in 2012, there have been a lot of other interesting studies, and we'll just run through them briefly here. There's been some very interesting work from England. Simon Heels and Robin Williams' labs have both published work looking at decanoic acid, which is a C10 fatty acid. This is highly prevalent in the medium-chain triglyceride diet that I mentioned a few minutes ago. And so the medium-chain triglyceride diet is it isn't more effective than the standard long-chain fatty acid ketogenic diet, but it's no worse either. It does have a slightly different side effect profile. It appears that decanoic acid has an impact on both mitochondrial function as well as on AMPA receptors, which is a subtype of glutamate receptors. There's also some work that's ongoing with epigenetics, so modifying gene expression with both the ketogenic diet and adenosine. Adenosine will have a downstream impact on methyl group donation. John Rowe and Do-Young Kim have looked at the effect of ketone bodies inhibiting mitochondrial transition pores. So this is thought to be a neuroprotective effect that's preventing ongoing seizures. And finally, there's a glucose analog called 2-deoxyglucose that's been examined. So this is not looking at the fatty acid side, this is looking at the glucose side. And it turns out there's just some recent work, beautiful work actually published in Epilepsy, looking at how 2-deoxyglucose increases tonic inhibition via extrasynaptic GABA-A neurosteroids. So we think of the GABA-A receptor as being a chloride channel that has unique binding sites for barbiturates and for benzodiazepines. It turns out it also has a unique binding site for neuroactive steroids. And it's the extrasynaptic neurosteroid binding sites that are actually thought to be probably more important than the synaptic ones. So that's some interesting work. There are some methodological issues, obviously, with all of these, but we decided when I was in Mike's lab, so this is now 2005, to take a different approach because we're seeing a lot of those lines crossing over and things were getting a little bit confusing. And Mike and I had a conversation in his office one day, and he said, you know, we're getting all twisted around here. Let's just take a step back and think about this, go back to first principles. So this is where our path to discovery starts. So we're going to dissect the anti-seizure mechanism using a pharmacological approach. We're going to treat the diet like it's a drug and see, so there's a standard battery, we're going to talk about this, of seizure tests that compounds go through before they sort of move on through the lab onto the approval process. It's a program that's sponsored by NIH. And then so we're going to subject the diet to those standard tests, then compare the diet to anti-seizure medicines that have known mechanisms. We'll talk about that. So if you have two things that look alike, then you say, well, the mechanism is probably similar. The reason why this approach was attractive to me is because instead of looking at something isolated in a dish that had been grown up in a culture under who knows what conditions, we're now looking back at the whole organism, whole circuit approach. So you're doing this truly as an in vivo experiment. That was attractive to me. And so, you know, this sounded so simple. So I'll tell you briefly about the classical test that we sort of thought about at the very beginning. So it's based on the NINDS, what's then was called the Anti-Convulsant Screening Project. It's now called the Epilepsy Therapy Screening Program. And so again, you can submit these compounds free of charge. You can submit a compound of interest free of charge to this program. And they will run the tests. The results are maintained confidentially, and so it's attractive for industry too, unless you agree to put it into a database, the so-called Panache database. So there are two tests I want to start off with. One is the maximal electroshock test, which identifies voltage-gated sodium channel effects. And so you would think something like phenytoin would do well in this. It's ocular and ear electrodes, ocular or ear electrodes. You give the animal the compound of interest, you subject them to the test, and then you see whether at different doses you can determine how much of that medicine you have to give for them to be protected. There's also the pentylene tetrazole test. This is a GABA-A antagonist, and it identifies GABAergic or T-type calcium channel effects. And that's a test that was typically given as an injection. So let me just sort of show you, I'm going to walk you through this because we're going to be using this series of tests in a few minutes. So phenytoin and lamotrigine both work in the maximal electroshock test, and they don't work in the pentylene tetrazole test. And they both act on sodium channels. In contrast, ethosuximide doesn't work in maximal electroshock. It does work in pentylene tetrazole, and that's thought to have a T-type calcium channel effect. And so if things match, they match. And if they don't match, then you think about a different mechanism. So that little conversation in Mike's office really is what started off this journey. So the problem is the ketogenic diet, other people have done this work, doesn't work in the maximal electroshock or pentylene tetrazole tests in mice. And so we needed to find a test that showed the diet effects in mice actually worked. And the reason we wanted to use mice instead of rats, some people have looked at rats too, and pentylene tetrazole works in rats. But if we wanted to do studies with, at that time, genetically modified animals, mice were the way to go. And I know people are doing rats now, but at that time it was really all about the mice. The other thing is that the diet can't be administered over a wide range of doses. And so drugs you can administer over 5 orders of magnitude in dose, and you can't do that with the diet. You can either go from 1 to 10 to 10 to 1, and you're going to kill the animals on either end of that spectrum. So in people, we typically use 3 to 1, meaning, so this is the ratio of grams of fat divided by the sum of grams of carbohydrate plus which is typically minimal, plus grams of protein. And so, so in humans, we typically use a 3-to-1 or 4-to-1 ratio. Modified Atkins diet is somewhere around 2 to 2.5 to 1. Typical American diet is about 1 to 5. So we love our carbs, and sometimes we love our protein too. So the problem is you can't do that in animals. We typically use a 6-to-1 or 8-to-1 diet. If you go to 10-to-1, you're going to kill them. So that's not an option. So we need to find an approach that doesn't require a change in doses. It's got to be something that's practical for the animal. And we want to do adapt tests for chronic diet administration. So we needed a new test in mice and a new testing approach. So there had been another electroshock test that had been described a few years prior to this called the 6 Hertz test. And the 6 Hertz test is another form of an electroshock test, but it's different in the type of, the way the current is delivered. And so rather than a sine wave, it's delivered as a square wave, and the settings are different in terms of how much current you're passing. And so it models forebrain seizures. It actually had been around since the 1950s. And then there was a drug that came along that did not work in maximal electroshock. It did not work in pentylene tetrazole. And the company that was developing it noticed that it had worked in a kindling paradigm. So this is a chronic seizure model. And they sort of started thinking about are there any other tests that we can take off the shelf, older tests that we can take off the shelf that might work? And so they tried their drug in the 6 Hertz test, and it worked. And it worked really well. Does anybody know what that drug is? We use it today. It's levetiracetam. So it was shown that it identified drugs that have different mechanisms. It's independent of pentylene tetrazole, and it's actually now replaced. So everything that pentylene tetrazole identifies as potential, the 6 Hertz test identifies. But not everything the 6 Hertz test identifies is identified by pentylene tetrazole. So the pentylene tetrazole test, the standard classic one, has now been dropped from the NINDS screening paradigm in favor of 6 Hertz. Okay, so the technique is, again, it's an electrode application. We do put local anesthetic on prior to the test. So what we showed is that the diet actually worked in the 6 Hertz test. And so what we're looking on the x-axis is the number of days on the diet, and on the y-axis we're looking at how much current was needed in order for that animal to have a seizure. And you can see around days 12 and 16 that there's a pretty big difference in how much current we needed to give a mouse that had been on the diet for 2 weeks a seizure compared to a mouse that had been on a regular diet. And so there are other seizure tests too that people have used, and we're going to come back to some of these. Chemoconvulsants, people, if you can mess around with a GABA A receptor or a glutamate receptor, someone has come up with a drug to do that. There are also kindling paradigms, and this is where you give a subconvulsive, subthreshold dose of current repeatedly, and then all of a sudden the animal starts having seizures. And then there are obviously a number of genetic models, which are interesting too. Okay, so when we did our ketogenic diet work, we showed that maximal electroshock didn't work. We're highlighted in yellow here. Maximal electroshock didn't work, pentylene tetrazole didn't work, 6 Hertz didn't work, and actually the canic acid test made the seizures in the diet animals worse, which was a surprise. So it looks, and we've got these long, you can look in one of our reviews, we've got this long comparison. The diet doesn't look like anything else in a whole variety of tests. We've done additional tests. We did adenosine, bicuculin, we did all sorts of stuff, and came up with nothing looked like the diet. So the diet has a distinct anti-seizure mechanism. So there are different ways now that we can sort of think about this and how to start breaking this down. So when we look at individual diet components, and we've already gone through this slide at the very top, people have looked at, we're going to sort of fast forward through this. Low carbohydrate does not mimic it. It's not the same as just a low carbohydrate diet. It's not the same as just a high fat diet that otherwise has a lot of protein in it. People have looked at polyunsaturated fats, at saturated fats. Nothing quite mimics the diet. And so we're going to move past that. I'm not going to show you the data for that. So the next question is, if the individual components don't really look like the diet, then what about changing? If you're doing a ketogenic diet, you're actually changing a bunch of things at the same time. So the diet is thought of as restricting certain types of calories, and it's a different kind of a feeding schedule. We'll get to the downstream metabolism pathways later. So the question is whether diets have, so as I said, the prior work showed that fat carbohydrate manipulation don't account for the diet's anti-seizure mechanisms. And so now we're going to try to change more than one thing at the same time. So for the students and the residents, where am I going next with this one? Any ideas? So we talked about how the diet was designed to mimic fasting. Okay, so that's why I kept saying that was my foreshadowing. So the expectation is if you put an animal on a ketogenic diet, or if you do a fasting regimen, those seizure tests are going to look the same in the animals, right? Because the diet's based on fasting, and the diet's all about fasting, right? Yes, okay, good. Turns out they're different. They're totally different. And this was a surprise. So this is work I did when I was in Marie Hardwick's lab. And there's a little bit of serendipity here in that we had, one of my mentors, I won't say who, once told me, well, you really thought this through and you really had this whole thing figured out and you knew the fasting was going to be different. And to be honest, it actually is based on a mistake that a lab tech had made in Mike's lab. And so when I was trying to reestablish the diet at Hopkins, I was thinking, what are we, something wasn't quite hitting it. And I said, remember that time when that lab technician messed up that whole batch of animals? And that was like our best batch of animals for the test. Okay, so let's take advantage of the mistake. So we have one group of animals that have been getting ketogenic diet for 2 weeks, and another group of animals where we're doing an intermittent fasting paradigm. So they get ad-lib feeds for 24 hours, and then we take their food out of the hopper. Then they get ad-lib feeds for 24 hours, and then we take it out of the hopper. So every other day they're getting ad-lib feeds, and every other day offset, they're getting nothing. They have water, free access to water the whole time. We did not dehydrate them. So we have normal chow, we have intermittent fasting, and the ketogenic diet. We also did a daily, for this particular experiment, a daily calorie restriction that was based on how much food the intermittent fasting guys were actually eating. So there's a little bit, I'm in there on the weekends trying to figure out how much did everybody actually eat yesterday. So what I'm showing you in the top graph is the 6 Hertz test, and so this is another way to show it. So these are basically you're used to seeing a dose response, so this is a current response relationship showing you increasing current on the x-axis. And you can see that it takes more current for a ketogenic diet-fed mouse to have a seizure in the 6 Hertz test than a normal ad-lib diet mouse, who's shown on the black line. And they were both, it turns out that the mice who were on either calorie restriction paradigm, it took less current, so they're more susceptible to 6 Hertz induced seizures. So the other question is, what about weight? And it turns out the 6 Hertz test, because it's an electroshock test, is independent of weight. And so everybody who was in any dietary manipulation group weighed the same on the day of testing. We didn't carry the daily restriction diet any further because it showed pretty much the same as the intermittent fasting. So here was the surprise. We did the canic acid test. So we basically inject canic acid. So the animals have been on their respective diets for 2 weeks roughly. We inject the canic acid, then you score behavioral scoring of seizures over the next 2 hours. And so a low score means you're not having seizures or just minimal seizures. A high score means you're having terrible seizures. And so what you can see is that the mice that were on the intermittent fasting diet actually did pretty well. And they got the canic acid was per weight. And the ketogenic diet guys did worse. So we've actually repeated this experiment using the same amount of canic acid. I'm not going to show you the data because they're unpublished, and we're writing it up now. But it turns out that it doesn't matter how much canic acid you use, the result's going to look pretty similar. And so one question, there have been other people who have looked at this, neuroprotection, intermittent fasting in adult rats preserving neuronal electrical integrity. And so this is where slices were taken out of hippocampi of intermittently fasted rats. You go ahead and you do the experiment and you discover someone else, has sort of looked at another aspect of this. That's always interesting. So we ended up carrying this one forward into the clinic. And so we were sitting in one of our little peds epilepsy meetings one day on a Tuesday. So that was the Tuesday at 11 meetings that we used to have. So that was our Tuesday at 11 meeting. And Eric Kossoff said to me, we should do this in the kids too. So we actually published a case series of 6 children who tried doing an intermittent fasting paradigm. We did what's now known as a 2+5 sort of a paradigm where they fast on 2 non-consecutive days in a week. And other people, there are other variations of this with limited caloric intake. People have been trying this for longevity too. But it turns out that some of the kids did show some efficacy. It was a tough diet. It was hard for them to continue it beyond 3 months. So it was a hard diet to do, but that's something that we keep sort of coming back to. And sometimes if someone's not doing well with the ketogenic diet, We're sort of kick-starting them a little bit with the fasting, with a brief fast. That's something that had been done for years. I didn't think of that, but they've been doing that for decades. But this intermittent fasting on top of that it works for some patients, and sometimes it sort of gets them back on track. It has to be supervised, though. We don't just say go off and do it, because you still have nutritional factors to follow and growth parameters. So just to summarize where we are, 6 Hertz and Kenic acid tests show a dissociation between diet and fasting. So I would argue, based on our data, that the diet is not exactly the same as fasting. But fasting itself has some value, certainly in rodents, and there's some efficacy in humans. Okay, so we're still looking for a mechanism, and we've found one really interesting intervention on this path, so we're going to keep going. So modifying fat or carbohydrates, not a mechanism. Intermittent fasting, not a mechanism. So we're going to go to the next layer of sophistication and say maybe we're altering some sort of a metabolism pathway, and that's what's going on. All right, so the master regulator of metabolism in the cell is the mTOR pathway with a mammalian target of rapamycin. And so I will talk you through this slide very quickly. The mTOR pathway has right in the middle, as you can see, TSC1 and 2. It's part of the mTORC1 complex, which should be familiar to you from tuberous sclerosis complex. It turns out that you can have a number of things influencing the pathway. There's upstream regulation by a gene called PTEN. Mutations in PTEN also can cause some overgrowth syndromes, just like tuberous sclerosis. It turns out that you can have alterations in glucose regulation and protein regulation, amino acids, that also impact the mTORC1 pathway. And then it got its name from a compound that inhibits function of the complex, and we're going to talk about that in a minute too. So just sort of proof of principle to show you that rapamycin works. This is work done by Mike Wong's group at WashU from a number of years ago. It's almost 9 years ago now, showing that rapamycin treats seizures in mice that have a conditional GFAP1 conditional knockout mice. So you can see the larger brain on the left and the smaller brain on the right. These are both affected mice. So the rapamycin limits the amount of growth. And as you can see in the lower panels, the mice that were knockout mice that were exposed to rapamycin did not have seizures in over the first 17 weeks of life, whereas those who received vehicle were having a ton of seizures within the first 9 weeks of life. There also was an effect on interictal activity. All right, so the bottom line is that rapamycin decreases mTOR activity. So we went back to our tests, and we actually, so this is one of those funny things about grant submissions, we actually had this figure in a grant application when that paper came out. So we sort of got scooped. And what it shows is that, what we showed in some blots, is that activity of the mTOR pathway is decreased in the ketogenic diet. And Mike Wong's group published a paper on this while we had this under review at NIH. And it turns out that there is a difference here. So remember, we had dissociation of 6 Hertz and Kenic acid tests. You can also see that animals that are, so these are mice, cortex, and hippocampus. You can see, particularly in the cortex, that we had, and we put a lot of protein on this, you can see that we actually had a decrease in activity of the mTOR pathway, whereas the intermittent fasting did not. And so we thought that was a potentially interesting thing to pursue. The study section did not agree, but that's okay. It got published by someone else. So Mike's group showed that the diet hypothesized, they actually didn't show, they hypothesized that the diet mediates its anti-seizure effects by inhibiting an mTOR pathway activity. So again, let's go back to our in vivo test, whole animal, whole circuit. So if the diet inhibits mTOR activity, if that's the mechanism, then an mTOR inhibitor should look like the diet does in these seizure tests, right? So it turns out we used rapamycin, and we couldn't get this stuff to work in the 6 Hertz test. Remember, the 6 Hertz test is where the diet is protective. We tried all sorts of different doses, exposure times, dosing regimens. We tried everything. My technician was sick and tired of it. He said, can we just stop these rapamycin experiments? So after 7 cohorts, I said, yes, you can stop these experiments. And you can see the number of mice that we used. And we were just sort of, it was a heavy-duty experiment, and we absolutely could not show any significant comparisons. What we did show was that after 3 days of exposure to rapamycin, at the very end of the monitoring period, there may have been some protection in the Kenic acid test. So that goes against. And those mice recovered. We didn't plot this out, but we followed them out for a few more hours, and that difference persisted. So we really didn't see anything that was substantial, certainly in the acute period with canic acid test, 6 Hertz test, we never showed anything. So the diet does not look like rapamycin. So let's take a step back. We're gonna sort of take another step back, go look at that mTOR pathway, and look at L-leucine. So L-leucine, it turns out, can increase mTOR pathway activity. So we're going to do a little pharmacology experiment and say, well, leucine should turn things on, whereas the diet and rapamycin turn things off. So this is one of those things that we wrote into a grant application that was sort of just, I call it like a dumb little experiment. We have to do it because it's a pharmacology thing. If you're going to turn something on, you have to turn it off. If you turn it off, you have to turn it on. And it turns out to be a very interesting experiment. So L-leucine, we'll talk about leucine for just a minute. It's highly abundant in mammals, most prevalent amino acid if you were to grind up a bunch of live tissue. High doses show some seizure efficacy in rats. Other people had shown this back in the 90s, both pentylene, tetrazole, and picotoxin, which affects GABA-A channels. But we know that high doses are toxic to humans and rodents. Think about maple syrup urine disease, where you have too much of the branched chain amino acids, and leucine is one of them. There also is a lot of potential for off-target effects, including transporters. Leucine is transported by a handful of transporters. There's endocrine effects that can have an impact on glucose metabolism, and we really don't understand its pharmacology. So it turns out that L-leucine protects against seizures. The other thing to note is that it's ketogenic. So it produces, it's one of 2 amino acids that is ketogenic only. So you're familiar, the students probably remember this. There's the glucogenic amino acids, there's the ketogenic amino acids, and then there's the amino acids that do both. So in this series of experiments, we did the canic acid test and showed that, shown in the purple line here, we sort of had to hit the sweet spot with the dose of L-leucine to be really protective, And in the 6 Hertz test, this is again a chronic exposure over 2 weeks. We showed that it took more current for a mouse treated with L-leucine to have a seizure than a mouse treated with just drinking its plain water, So L-leucine protects against seizures. It looks like it works in both tests. So now we're sort of thinking maybe we're onto something with this ketogenesis. So remember, not all amino acids are created equal. So the question was, why did we see these results? These are kind of funny results. If it's really ketogenic, then you would expect it would do worse in the canic acid test, not better. And that didn't make sense to us. And its exposure, but it also worked in the 6 Hertz test. So the question is there something else? Is there something about the L-leucine production? Is there something about its transporter effects? Are we dealing with some sort of an off-target effect? Are we dealing with some sort of a contaminant? So my mentor, Marie Hardwick, and I went around and around about a contaminant, and I was talking to other people outside the lab about transporters. So it turns out that D-leucine, so have you guys seen that meme on Facebook where it's Samuel L. Jackson versus Samuel D. Jackson? Yes, it's a biology joke. Okay. The students and the residents aren't even using Facebook anymore. So, so D-leucine is found in plants and in the microbiome and in beer. The beer part's really fun. It's present in the brain, including the hippocampus, so it's relevant to seizure generating regions. Its role in mammals is not defined, and it has never been studied in epilepsy. So I'll tell you about the beer in a minute. We're going to come back to that. It turns out that a scientist named Gustav Emden had studied D-leucine in liver preparations back in 1906 and 1908. I love some of this history stuff. Does anybody know what Emden is famous for? He's famous for a couple of things, but really big famous thing. Anybody familiar with the Emden pathway? Has anyone heard of glycolysis? That's the Emden pathway. Okay, so even this guy was trying to figure this out, and it turns out that D-leucine is more ketogenic in the liver preparation in the dish than L-leucine is. So we had one of my former residents, who's now a faculty member at Hopkins, who's from Germany, translate this for me, and it was actually kind of fun sitting there over a cup of coffee, where she was just she was almost more animated and excited about it than I was, but it's really cool. So maybe we're onto something with this ketogenesis. So we tried D-leucine. You can actually buy D-leucine. And look at what happened in the canic acid test. So we had 2 doses that worked in the canic acid test really well. It also worked in the 6 Hertz test. Turns out we looked at the amount of contamination in the supply, that wasn't it, and this obviously answers our transporter question. So now in our path to discovery, we have yet another, in our path for search for a mechanism, we have another path to discovery. And so now de-leucine, so this looks interesting. And the university was interested in this for a while. So you can ask, is this just because it's producing ketone bodies? And so we're looking in the figure on the left at the canic acid test for L-lysine, which is the only other ketogenic only amino acid. I know the students remember that it was leucine and lysine. I know you guys have that memorized. So we looked at the other one, and it turns out that didn't do, yes, it was statistically significantly different, but as I was saying to my mentor at one point, if I were a veterinarian treating mice with seizures, I would not use L-lysine for treating that mouse's seizures. We also looked at blood levels of beta-hydroxybutyrate and showed no difference for the L-leucine cohort. And so we don't think that this is a ketone body mediated effect. But more important, as a clinician, I said, you know, we don't go around treating everybody with seizure medicines before they have their first seizure. We treat them after their first seizure. So what happens if we give the medicine after the first seizure? And people really haven't done this a lot. And so it turns out we gave the de-leucine, and although there was a little more prolonged lag in the onset of effect, we actually showed that there was a difference between the groups, even when they're treated afterwards. And so one day I have this crazy idea, and I said to my technician, I really want to amp it up. Let's give them even more canic acid, and let's compare it to diazepam, because we have to compare it to something. Because we had started talking at this point to some companies, and they said, well, how do we know what you're using is any good? So we have an experiment here, a canic acid test again. They've been injected, so they have not been injected with anything. These are naive mice who are injected with canic acid at time 0, and then they're having their seizures. You can see their seizure scores are increasing, and then we give either de-leucine or diazepam, or a combination of the two. So the mice on the black line on top were treated with the respective vehicles. The mice in the green line were treated with de-leucine. The mice in the blue were treated with diazepam, and the mice on the red here were treated with both. And what you can see is that the de-leucine mice returned to normal baseline activity sooner than mice in any other cohort. It took another hour for the mice treated with diazepam only to return back to baseline. It took probably another 10-15 minutes for the combination mice to return. So by baseline activity, we mean they're sort of sniffing around, hanging out with their cage mates and litter mates. And then we said, well, so we had to sort of backtrack a little bit. Does L-leucine work after seizure onset? And the answer is no, it doesn't work. All right, so we published these data a couple of years ago. So when we talk about epilepsy therapy, all the drugs that we're using so far have been evaluated using acutely induced seizures, and I've shown you only acute seizure data. So more emphasis is now on treating recurrent unprovoked seizures. So the other question is, could de-leucine be inducing some sort of a disease modification effect or a chronic effect? So I'm going to show you a series of experiments with male FBB mice who were 5 weeks when we implanted electrodes. So basically we implanted electrodes, exposed them to canic acid, status epilepticus. So we kept giving them canic acid until they were just persistently spiking in status overnight. We were hydrating them, obviously. And then the seizures stopped the next day. We gave them leucine, so we had a 1 month period where they were off drugs. They would just really make sure their seizures are established. Then 1 month on drug, and then took the drug away, and another month off drug. So we've monitored these mice for 3 months at a shot. And so what I'm showing you in this slide is just an EEG representation of what their seizures look like on the tracing. So these, I'm finishing up this manuscript right now. We're about to submit this. So we were talking to our statistician. So here's another lesson I want the students and residents to take away. Talk to your statistician before you design an experiment, because they will help you figure out whether you need to modify your design, or they may have different ideas. And so I said to our statistician, Carol Thompson, up at the Bloomberg School of Public Health at Hopkins, I said, Carol, I want to set this thing up. And I gave her some data, and she said, you should look at the difference between day and night. I said, well, that's kind of silly, because I really don't care. She said, well, no, you should care. And so it turns out Carol had a great idea. So what I'm showing you here is 4 of the mice. The 5th mouse looks similar. And so in the shaded box to the left is when they're off treatment. and then the shaded box to the right is when they're off treatment. So what you're looking at is whether there's a difference between the treatment and the post-treatment period. So we have this mouse who looks like he probably didn't have much of a response. We have this mouse who looks like maybe, maybe not. We have this mouse who had a fantastic, you call this one a super responder. And then you had this mouse where things may have actually even gotten worse. So we looked at all 5 mice together, and this may be a little bit difficult to read. But the bottom line is when we look at overall over time, the D-leucine didn't make any difference between the 5 mice. So that was kind of disappointing. The seizures did get worse over time, but then we looked at Carroll's analysis, and Carroll this is Carroll's idea. It turns out that mice, when we look at the pre-treatment versus treatment period, it turns out that the mice had fewer seizures on treatment during the dark cycle. So that was pretty interesting. So why is that biologically interesting? Because D-leucine, I didn't tell you this, is highly expressed in the pineal gland. It secretes melatonin. So we need to think, I would posit that we need to think a little bit differently about how we're evaluating seizure drugs. So we sort of moved away from this ketogenic diet stuff. Again, this is sort of a discovery pathway. During the light cycle, there's a difference in seizure frequency on the days when the mice had seizures. And the interpreter is actually thinking a lot about this because, like I said, we're writing it up and I'm about to send it off, and I'm sort of rethinking my interpretation. It turns out that the mice had a lot of seizures in their post-treatment period, and so there may be some efficacy during the light cycle and seizure frequency, but I can't pound my fist about it and say this is a definite thing. So we had a decrease in the number of nights with seizures during the dark cycle, no change in frequency. When they did occur, there's no change in frequency. There's a sort of questionable finding during the light cycle. Clearly, there's no effect on disease modification, and I think that we need, as we evaluate new seizure compounds, I would argue that we need to think about doing this kind of experiment more often. It could be that we started the treatment too late and we started it earlier in epileptogenesis, we would have seen fewer seizures, but latent periods in mice can be highly variable, as we saw with our experiment. So it's a tough experiment to do, but I would say that if there's any effect, it's an anti-seizure effect, and it's a limited one. It's not an anti-epileptic effect. So I have not, much to my disappointment, I have not found the holy grail here. So implications for mechanisms, what does this mean? So D-leucine is highly concentrated in the pineal gland. So I would argue that it may be an interesting tool to use to look at circadian seizure patterns. So there's a lot of work that's been done for almost a century, a little more actually, looking at differences in seizure frequency during different phases of sleep. And seizures have an impact on sleep duration. So switching gears a little bit, we're still looking for a receptor. And what I didn't show you is that we did a screen through NIMH looking to see if the D-leucine bound to any of the known major receptors that are in this NIMH screen. We did a screen at NIDA, nothing turned up. So it turns out there had been a paper that had slipped my initial search. PubMed's good, but it's not phenomenal. And I did a search on another engine and came up with a paper showing that D-leucine actually binds to taste receptors. So the D amino acids, it turns out, are sweet. So we're back to eating, all right? It's got to be about the food. So let's talk about taste receptors for just a minute. It turns out that there's a class of receptors. These are G protein coupled receptors that can form in different subunit assemblies. The umami taste is the so-called R1, R3 combination. The sweet taste is the R2, R3 combination. And D amino acids, when you taste them, are sweet. And we actually showed that our mice had a slight increase in how much water they drank while they were on treatment compared to both before and after. So that was interesting. And when you taste, so I'll admit, I taste a little bit of it myself. It's pretty sweet. The L amino acids are not sweet. They're either neutral or they sometimes can be a little bit bitter. So the taste receptors were not in the NIMH or the NIDA screens. So we acquired some mice. So here's the data published in 2014. This is all evolving, showing that, in fact, the D-leucine is a ligand for this particular receptor, whereas the L-leucine is not. So we acquired these mice from Pablo Ortiz at Henry Ford. These are mice that Charles Zucker actually created when he was at UCSD. He's a Howard Hughes investigator. So we acquired these mice, and we said, well, okay, well, let's just the basic experiment. Let's just see what happens with the knockouts compared to the wild-type littermates. And it turns out that these mice are protected against maximal electroshock seizures. So that's pretty interesting. We don't know what D-leucine actually does at this receptor. We don't know whether D-leucine is responsible for that. Other people had actually shown that certain ligands, when they bind to the taste receptors, this particular receptor, are protective in the maximal electroshock too, which wouldn't make sense if you're talking about a knockout. And we think it may be, there's a couple of reasons. One of it may be that we're looking at an inhibitory effect rather than an excitatory effect. We'll skip that slide. It turns out it didn't work in the 6 Hertz test either. So other applications, we're going to sort of generalize now. Other applications of metabolism, so one thing I didn't tell you, taste receptors are expressed both gene and protein in the hippocampus and the cortex. So that's where we think things are actually happening. Okay, so other applications of metabolism-based treatments. It turns out that when you look at some of the original work in the ketogenic diet, it was done for urinary tract infections because it changes pH in the urine. Enzyme transporter deficiencies, as you might imagine, using a ketogenic diet in particular as a way to bypass a metabolism block. People are publishing work on brain tumors, cerebral ischemia in rodents, dementia, cognitive impairment, Kabuki syndrome, Parkinson's disease, ALS. So this is a spectrum of work that's been done both in vivo and in vitro for different paradigms. So we are going to conclude, and I guess the really main point I want to make is that metabolism should be thought of as an option for treating seizures. We've talked about the ketogenic diet, intermittent fasting. I briefly described some work on 2-deoxyglucose. I didn't show you any data for the low glucose, the low glycemic index treatment, and I haven't shown you the work on fructose 1,6-bisphosphate or some of the anaplerotic substrates, but there are a number of different points in the metabolic pathways, so hopefully this brings some of the biochemistry home, where messing up the metabolism pathway or substrates may induce or may confer an anti-seizure and possibly anti-epileptic effect. So on our search for mechanism, we had a lot of interesting little side paths to discovery. We never found the mechanism. I think it's safe to say that the ketogenic diet and other metabolism-based treatments have a number of mechanisms, just like a number of drugs do. As we learn more about the drugs, we're finding out the same thing about the drugs. So with that, I wanted to thank my mentors along the way, my technicians and my clinical mentors, as well as my funding sources, NINDS, the Hopkins School of Medicine, the Pakula family, the Maryland State TEDCO was supportive, as well as the Hopkins Veterans Health Institute and my collaborators. So I guess here's the slide about claiming your CME for your login and recording your attendance. The text code to text is 9642. And I think, yep, we're going to leave it at that. Great. Thank you very much for your attention. I guess we have a few minutes for questions.
Yes. Yeah. You want to use the
Mr. Microphone.
Thank you. Yeah.
Hi. So I find the intermittent fasting very interesting. So you said 2 days of non-consecutive days per week. How short can the duration be? Can it be 16:8? That's pretty doable. And the way I know about it, it's a big weight loss fad these days. But 16 it's hour by hour, or 16 hours fasting, 8 hours eating, that kind of window, which is cycle daily. Does that work? Because it sounds interesting because it's sort of protective after acute seizures. That's
So the question is, can you alter how much can you alter that fasting schedule?
Yeah.
And the answer is we really don't know. We had to get this through our IRB, and we sort of started with an agreement. We would do sort of 2+5.
Yeah.
It's now being called 2+5. We sort of said 2 on, 5 off.
Yeah.
We're talking about growing children. As a pediatrician, it's got to be safe.
Yeah.
So I don't know, with an adult, you probably could.
Yeah, adults usually, there's a lot of, there's no literature, but there's a lot of media or whatever. It's about 16:8 or hour by hour, which is cycled every day. So that's sort of interesting.
So the longevity people have looked at this
Yeah.
a lot more thoroughly than we have.
Yeah.
I don't know, it's certainly, I think, very interesting.
Yeah,
but
it is.
There's a lot of work that needs to be done before I can prescribe it like that. I put my physician hat on too. And you want to advocate for the treatment, but it's got to be safe, right? So the answer is I don't know. We didn't look at it because we're dealing with growing children. And nobody's growth is affected in the short term.
Thank you.
Adam, thank you very much for the talk, and congratulations on maintaining this very inspiring path of focusing on one subject and getting serendipitous and non-serendipitous great findings. I have a question about how do you explain? I have a couple of questions actually. First, how do you explain that D-leucine can achieve seizure freedom or seizure reduction faster than D-leucine plus diazepam?
So it's a good question. So the question is, why did D-leucine do better by itself than the combination of D-leucine and diazepam? So these were behavioral seizures we were measuring, and so I suspect that there was a sedative effect of the diazepam that hadn't worn off yet. That's my guess. When we do months, we did do some monitoring on a handful of months, and it looked like there wasn't a huge difference in the amount of electrographic seizure control. The D-leucine guys are up around the clock.
Sure. Yeah, that makes sense. Do you do some experiments with intracranial monitoring with hippocampal?
So we did not do that. That was a whole.
Yeah.
So we were getting ready to do that.
Okay, gotcha.
Technically, that's actually.
Sure.
We would love to do it. But it's in our protocol.
Perhaps we can collaborate on that. We're doing it here.
Oh, good.
Yeah. So does melatonin increase with D-leucine? Do you know?
Fantastic question. We don't. And we don't know what happens at baseline. We don't know what happens in the muscle receptor. But we would love to know what happens in melatonin. Both in the blood and in the brain. So we're talking about the beer story. So D-leucine is discovered in beer by scientists with great chemistry and strong intentions. And he published this paper back in the early 90s, and he was looking for someone to actually look at tissue, not D-leucine tissue, fascinating. And so I emailed him, and it turns out he's a collaborator. We just had a paper in the American Chemical Society that was looking at concentrations in mice that are untreated for D-leucine. And other DNA acids that happens to publish about 10 or 15 years ago now. It turns out there's some new analytical methods that actually are sensitive. And so now I've spent time actually making that up in Baltimore. So we'd like to be able to measure that. It's, pale is very small, and it's a very tricky assay to
non-alcoholic beer is not sedating. Is not nearly as sedating as alcoholic beer, so I don't know if melatonin. Okay, I have one last question. If nobody else does, do you have questions? Yeah.
So for the ketogenic diet, mostly using kids, pediatric population, certainly very rarely in adults. Is that more like the practical reason, or you feel like the pediatric and the adult population, the metabolic will be different?
Yeah, so that's a great question. I'll repeat it. So this is really important. The question is whether the ketogenic diet has different efficacy in adults versus children, because you hear about it being used in children more than you do in adults. And it turns out people have actually looked at this. So Joe Survey actually had that back in the 90s. I think people looked at it. So when the adults can stay on the diet, the efficacy is the same as children. I think adults have a little more of a problem because we have, going back to the taste receptors, we have a much more mature eating experience than they do. And so I think I would suspect their cravings are a little bit different. So I think it's harder for adults to do it. But when they do it, they do just as well. So I don't think it's a difference in metabolism.
And of all those animal models and those tests like mice or possible rats, and are those tests like on ketogenic diet, they are doing the young, like rodents or like?
So the question is, what happens? So how have the rodent studies been done? The vast majority of studies have been done in what we call sort of adolescent. it's very hard to get the diet in reliably in a mouse or rat that hasn't moved yet. And so all of our experiments are done post-weaning mice, so we start at 3.5 weeks. So there are some groups that have looked at the effect of the diet on aging. And so there are some experiments in that group.
We have to empty the room, but I have a burning question, which will be the last. Which, in your experience, is there an anti-seizure medication or medications that work best, most synergistically with the diet?
So my colleague Eric Kossoff looked at this, and the number of patients is super low. Eric's conclusion, I was on the stage, Eric's conclusion is that's in slide work. I think that there's some methodological issues, but if you work, I'd love to see something else. Do you guys want to ask that question? Sort of too much. It's a great question. We really think that's something you guys have done on we've worked that well, especially with these early studies. The kids are all on calcium,